Good day everyone, I am ALTHEA MAE Olasiman and I am the first reporter. So today, we will be discuss the chapter 11 which is gene therapy. Diba, in the previous chapter, we learned about the nanoworld which is contains atoms and molecules. So in this chapter, we will focus on the developments in the nanoworld as applied the animate or the living, especially in human health. So what is gene therapy? So gene therapy is a treatment or cure for disorders caused by mutated genes. It involves adding a normally functions a copy of a gene to affected cells that restore the normal function. So this means that instead of using the regular medicine, so gene therapy focus on fixing the problem at the genetic level. So when a gene becomes damaged or mutated, so the body cannot produce the correct protein needed for normal function. So by inserting a healthy gene therapy helps the body work properly again. So gene therapy is different from traditional treatments. The main goal of gene therapy is the root cause of the disease, not just the symptoms. So he is Wilson Fringe Anderson. So he is called as the father of gene therapy because he was the first to successfully apply gene therapy in humans. So in 1990, he treated a 4-year-old girl with adenosine or deaminase or ADA deficiency, which is a rare genetic disorder that prevents her body from making a certain immune cells. So he didn't just as an experiment. He proved that the genes could be safely modified in human cells that treat diseases. So his work laid the foundation for the later treatments for genetic disorders, cancers, and even viral diseases. Although gene therapy is a promising treatment option for a number of diseases, including inherited disorders, some cancers, and certain viral infections, the technique remains risky and it's still under study to make sure that it will be safe and effective. So ang gene therapy naa gihapon siya ay potential nga makaayo og mga serious diseases. For example, genetic disorders like hemophilia, some certain types of cancer, and even some viral infections. However, gene therapy is still new, and because of that, scientists are still studying it to make sure that it is safe for humans and that gene therapy is safe. So gene therapy is under study to determine whether it could be used to treat diseases. So current research is evaluating its safety, and future studies then will test whether it is effective. So this means that experts are still trying to understand how safe gene therapy really is. So although many patients have shown their improvement, there are still risks such as inflammation, immune reaction, and even cancer. So this risk happen because adding genes into cells can sometimes affect other parts of the DNA. However, scientists are continually improving the technology. Then modern vectors are designed to be safer, and clinical trials are carefully monitored to ensure to their officials to ensure that the highest level of patients' safety. So the process of gene therapy. So the process of gene therapy is it involves using a vector. So usually a modified virus to deliver a new genes into a patient's cell. So first, so the new gene is like a set of instructions. So it tells the cell how to make the right protein and to fix the broken problem. So in step 2, which is the virus vector. So the new gene can't move on its own. So it jumps into a virus vector. So the virus is made safe and works like a delivery truck. So the job of a virus vector is mahatod niya ang new gene into the cell. And step 3 binds to the cell. So the virus, ang virus ani, the vector finds the cell and sticks to its surface. So this is like kanang rag, kanang nanukto ka sa pultahan. And the cell will know if naabay tao nga padulong or nice something. So if dili mo stick ang kanang cell, so the gene can get inside. Step 4, vector packaged into the vesicle. So the cell, arina ang cell wants to take the virus inside safely. So it's wrapped in a small bubble called a vesicle. So para madaan niya nga safety. So once nga na siya sa is the step 5, vesicle breaks, releasing vector. So once na siya sa sulod, the bubble opens and release the virus. Now the virus is free to move around inside the cell. So this step, it is very important because the virus needs to reach the nucleus, which is the control center. So ang then after na ito sa cytoplasm. So the virus travels through the cytoplasm, which is the space inside the cell but outside the nucleus. So mura siya ganang, so di ba the virus moves carefully, so that the virus moves carefully through the pathway, so it delivers the gene safely. Then after that, ato siya padungsa, which is the process the step 7. So the nucleus is the control center of the cell. So it holds all the cell's DNA and instructions. So the new gene must reach these control centers to do its job. So pag sa number 8, the new gene injected na into the nucleus. So katong virus nga deliver na new gene into the nucleus. So the cell can read the new instructions and make the correct protein. So kani ma-affects ng ilang problem caused by the faulty gene, and it helps them the cell work normally again. And there are two types of gene therapy, which is somatic and germline gene therapy. So first, let's start with somatic gene therapy. So according to this type of gene therapy, it involves transferring a section of DNA to any cell of the body that does not produce a sperm or eggs. So this means that the therapy only affects the patient's body cells, like skin cells, liver cells rather, or blood cells. So the changes happen only in their body sa katong patient, and these changes will not be passed on their future children. For example, someone has a genetic blood disorder, so we can add a healthy gene to their blood cells, so it help the patients, but their children will not inherit that changes. As you can see on the image, the only a patch or part of the body has the mutation. So the sperm or egg cells are not affected. So none of the children will carry the mutation. ikaduha is germline gene therapy. So it is transferred of the DNA to the cells that produce sperm. So these cells are responsible for making future babies. So any changes in these cells becomes part of the patient's children as well. So this means the corrected gene will be passed on the next generations and even the future generations after that. You can see that the mutation is in the egg or sperm cell. So when the embryo develops, the entire organism carries the mutation. So half of its sperm or egg cells also carry the mutation, meaning it can be inherited by its children. So in simple words, somatic gene therapy affects only the person that receive the treatment or receiving the treatment. While sa germline gene therapy, it only affects the person and their future generation. And that's all. Thank you.